Hi there, everyone. Thanks for joining us through that last session of the day, and looking forward to a panel discussion here as part of session 4. I think in the goal of session 4 was being able to hear a little bit more on the details, maybe put in some provocative ideas interspliced, and then through the course of the panel discussion, be able to logically reference those talks that weren't necessarily a part of this session, but have certainly helped educate us all to what we've listened to over the last couple of days. So in that effort, and Donna, thinking about your talk that was up first, I think it gave a great summary of a lot of the themes that came up through the conversation, but also put it very nicely into the regulatory framework that reviewers at the FDA and other health authorities find themselves in, which is coming to benefit risk decisions. And specifically when considering some of the aspects with regard to the incorporation of PEDS within these studies, and we think about type 1 diabetes as a disease, recognizing that the kids who get diagnosed are going to have to live with the disease the longest and potentially have the longest burden on their life. When we think about that as just as to how it is, and we start to overlay it with some of the considerations that you went through on the subpart D description, which thank you for covering that, and the concept of minor increase over the minimal risk, with the need to be able to establish a direct benefit. I certainly see overlaps in the conversation when we're describing that clinical benefit through things like accelerated approval. So I realize that's a bit of a long-winded explanation, but up the top. But to get to the question, when we think about it in the hypothetical situation of agents that are being brought forward kind of de novo, and ones that would be being repurposed from other potentially immunosuppressive indications, and realizing this is hypothetical, but how does that weight in the balance? And I know it would depend upon the certain safety profiles, but just to get a little bit of perspective on that between those two different types of agents.
So is your question?
Sorry.
Can you hear me? Can you hear me?
Yes, we can. Yeah.
Okay. Yeah. Great. So I'm not quite sure I completely understand the question, but what I think you're asking is how do we justify doing early phase 2 trials in children when we haven't determined that C-peptide is a validated surrogate? Is that part of the question that you're asking?
Yeah, I think that was better phrased than I put it.
Okay. So, if you look at what I went through in terms of prospect of direct benefit and the benefit and risk calculations that we do in pediatrics, when we're doing a study in children, we don't, the level of evidence that we require to support a prospect of direct benefit is not as great to conduct a pediatric trial as we might require for regulatory approval. So this change in C-peptide that you're seeing in these, when we look at the mechanism of action of some of these products, would be weighed against the risk of the child's exposure to that product. And we have allowed studies in the phase 2 space to move forward where we've looked at C-peptide as an outcome in a trial with the idea that, information from that trial will lead potentially to a phase 3 trial where we might be able to look at clinical outcomes that might support regulatory approval. And as I mentioned on one of my slides, to support prospect of direct benefit, we don't always require adult data to support that. We require, we may use non-clinical data. In this case, we have some adult clinical data to support that, and adult data in children to support that. There's a change in C-peptide when you expose individuals to some of these products. So I think that's enough to move forward with pediatric studies. And again, it doesn't rise to the same level of evidence that you might need for product approval.
Maybe if I can jump in, relative to Europe, would that be a very similar sort of thought, or what would your opinions be regarding that, Peter or?
So I think the trial and the duration of it should still be in the realm that we both understand what it does, at least on the C-peptide. And I still think it's important to really have some grasp of something that is more clinically meaningful than that, at least at this stage for us really to see, because we will need to have that 2 years, I think, also to look at the safety and many of the drugs that are also shown in the presentation earlier by the statistical colleague. I think there we really saw that these are no sweets, these are no candies. They really have also on, especially if we go to the younger children where the immune system is not yet fully settled. We don't know what the impact will be on the long term. So I feel that I still require something that shows what could this treatment do in the long term, and what do I already have to in the duration of the trial really to tell the parents of these children what their children are experiencing when taking this drug.
So can I add something to that?
Yeah,
No, I
please.
think
Donna. Go ahead.
No, go forward, please.
So I was just going to say that that generally these trials would also be of sufficient duration where we would think that we might see some clinical benefit within the context of that study, but it might not necessarily be, again, a clinical benefit that we might use for regulatory approval. We do look at that, as I mentioned, when we're looking at prospect of direct benefit. We want the dose to be adequate, and we want the duration of exposure to be adequate to have some potential benefit to those children who are participating in that trial.
Okay, thanks. I think there were some issues that were brought up related to the use of control groups, and obviously that's one of the factors that I think you brought up as well, Donna, and also T brought up in terms of how we can incorporate that, minimize the risks of the control group, if you will, particularly, again, as we think about pediatrics. I think T threw out a few ideas there relative to ways to minimize risk. T, if you want to bring that up and see what you think, moving that forward into the clinical trial space we're talking about. And then maybe if others could react to that relative to the data that, this cumulative analysis sort of suggests that would give us a comparator to look at.
Yeah, comment was really partly to just give folks the best chance of receiving potential benefit of a therapy under the assumption that the therapy would have a benefit. So, of course, minimizing risk for placebos. There's not a lot of risk in placebos, of course, but if there are risks in the trial, then having less subjects exposed to placebo, if there is a risk, would be of course, a positive thing. But the real concept was that C-peptide decline is very predictable, and do we really continue to need sugar pills to study this decline in placebos when we have a really pretty precise and confident expectation of how fast C-peptide will decline 1 year after diagnosis, and can instead we just expose participants to potential therapies and then compare it against a massive amount of data that we do have to make judgments on whether a therapy has promise for early stage study.
And I think the risk of control in terms of not getting therapy in the fall and why it's hard to recruit trials is really what I was driving at. I think, Peter?
Yeah, well, I think that the analogy to the oncology I do understand. And I do understand that the reliance then really in a single arm trial, you really would have to look at the C-peptide on its own. You can't really interpret the clinical outcomes anymore that may not, for which the study may or may not be powered, or actually I don't think it would be powered after having seen the presentations. But also the difference with the oncology setting is perhaps that there the adverse events may be less immediately challenging the patient, because we're often there in a setting where you haven't got any other treatment options. Here, these children will all be concurrently treated with insulin and well controlled with that. So I think a single arm trial would be really difficult to defend in my perspective at least. It may give you an insight on C-peptide, but that's not going to help me in making a decision.
Hi, everyone. Jackson here, a latecomer to the game, but a couple comments on borrowing of historical controls to augment placebo arms or even replace them. There was a pretty rich workshop right before COVID started, sponsored by DAA with the Office of Biostatistics, FDA heavily weighing in. And a central theme was the statistical considerations for borrowing of historical controls. And some of the main themes that came out of that meeting was really an articulation of a double-edged sword regarding the borrowing. So the big concept that came through was understanding the congruency between the controls that you're borrowing from and the contemporary active arm population. And there were a lot of differences highlighted, or a lot of key topics highlighted in that regarding the eligibility criteria, the treatment procedure, the facilities, the care providers, and just any differences in the standard of care over time. And then there were an additional set of considerations on the statistical side, where the statistical machinery that you need to borrow the controls, which are often set in a Bayesian statistical light, have some assumptions that can cause major inflations of errors, can reduce power, and can actually completely confound what you think you're looking at. If these considerations are really put carefully in the right context and well understood, there may be conversations that could advance with the regulators, but there are a lot of big hurdles that need to be addressed both on the clinical side and the statistical side. So I just wanted to put in that quick comment there.
Yeah, I mean, these methods are used in many different settings. Any method, needs to be put into the context of its use, but certainly Bayesian methods and methods that, or frequentist methods, can be used in this context. And I don't see or at least I'm not aware of the big limitations of using these kind of methods in this therapeutic setting versus, the dozens of other therapeutic settings that they're implemented in. We do have a lot of data that's showing very, very similar things across multiple studies, different ages, different baseline C-peptide values. The predictability is pretty good in the decline of C-peptide.
Yes, and just to that. Francis, please go for it.
No, I was gonna change topic, so please go ahead.
Yeah, just on that one as well. I think one of the parts that we see at c-path quite frequently across multiple indications is the body and field of research that can be in the public domain through academic conferences and also through publications. The opportunity to be able to put that information in front of regulators in an anonymized patient level format can be enlightening to help forward conversations as well. So T, when you talk about the predictability and basically from the world of TrialNet and the great experience you guys have, mechanisms with which to be able to make that and more accessible to the folks to vet it in a way that's more in line with regulatory decision making is just an aside I'd put onto that as well, not disputing the validity of it in any setting, but rather the ability to have the reviewers at agencies do their reviewing job. And Francisco, I pass it over to yourself.
Before we move on, I'll just say from an FDA perspective, I think it is accurate to say we're open to this concept, although it hasn't been applied, to my knowledge, very broadly. We've seen it suggested for pediatric type 2 diabetes, where I think it's a lot more challenging. You have to be sure that the patients in the natural history cohort are similar to the ones that you would be treating in the trial. And for type 2, we just don't think that that's generally feasible, because for type 2, it seems that the kids who enroll in trials are just very different than the ones who don't come to medical attention or in the real world setting. So that's been a huge challenge. But for type 1, you know, I'm not completely convinced that there isn't something here. I think I personally have a slightly different view than Peter about the idea of insulin being an acceptable therapy. I see this as a disease where there's an opportunity to intervene before beta cell function is completely gone, and it's not the same as just, you know, giving a therapy for glycemic control in the type 2 setting, where if you don't do something for a while and put a kid on placebo for 6 months, that's reasonable. But here, I have a lot of trouble with thinking about if we really do think a drug could be a disease modifying, make a disease modifying impact. It's hard for me to not try to maybe work a little bit harder to see if we can find ways to minimize exposure to placebo in this particular disease space, despite the statistical challenges. So I think that we would like to see more, although I can't comment on any specific methodology today.
Peter.
So maybe I do see.
Thanks for that, Lisa.
Sure.
Oh no, Peter wanted to respond.
Yeah, I want to respond because I think my point is not completely that I say that insulin is the perfect treatment. I think I absolutely agree that it has its shortcomings. I think especially also the stories of the patients of today were again very compelling in the way that all the things you need to do, and even if you have the device that gives you insulin based on your CGM measures, if something gets detached and the problems around that and the problems with the hypos that they experience, that is clear. I mean, but these treatments are there, and especially in the initial stage, we don't see too many of the issues yet with that. So I think it's really important that we get at the stage of the development of new metric, we really need to try and link it prospectively with the interventions that we're trying to test what happens, and that it's not just this C-peptide in the correlation studies that shows this promise, but it also is independent of the different interventions. And for me, that is at this stage why I'm more concerned. Mm-hmm. And I think in that realm that we need to have further discussions also.
Yeah, I mean, you raised the point of, I agree with you about C-peptide and what its current value is in this setting of drug development here. And you had made a statement that we really could only consider these types of single arm approaches once we have validated C-peptide. I think that's what you were implying. And if that is actually logistically accurate, I'd have to think that through a little bit. Then I think that we have big challenges about trying to utilize that approach right now, because we do need those clinical outcomes. But if there was a way to minimize exposure to placebo and still maintaining the paradigm of C-peptide as a intermediate endpoint or reasonably likely surrogate with the requirement to obtain data on clinical outcomes, maybe that's not possible, but that's more of what I was thinking.
Great. I think just as a note, I think I have a big time delay on my side, so apologies if it's slightly disjointed. I'm cutting across you guys, but Francisco, you had a point you wanted to raise there.
Yes, thank you. Actually, I was going to link the conversation on placebos, borrowing placebos, to Dr. Bergold's communication to us. That was a very impactful message. That Novo Nordisk thinks that clinical trials in T1D, phase 3 trials, are basically almost unfeasible if glycemic control is to be a primary endpoint. So I just wanted to go back to that, to the feasibility and the practicality of the studies, because that's what we we're going to discuss in this couple of days the use of c-peptide as primary endpoint or not. And by way of introduction, I was not a panelist, so just quickly, I'm the Chief Scientific Officer of Provention Bio, and we are a small biotech company that is continuing the work done over the past 2 decades by many of you with teplizumab. And now we are in conversations with FDA and EMA on bringing teplizumab for the delay of the progression of clinical T1D, as well as we have a phase 3 trial in newly diagnosed patients. C-peptide is very important for us as supporting evidence in stage 2 T1D and is our primary endpoint in the phase 3 trial in newly diagnosed. So what we wanted to share with you was just our view is maybe the view of the small company, a very pragmatic view. And as Dr. Janov said, we need to find the balance because patients are waiting. So just wanted to go back to the topic of C-peptide. And I hope we can all agree on a minimum set of principles over these past 2 days. T1D is primarily driven by autoimmune destruction of beta cells, resulting in loss of insulin, which leads to all the metabolic and derived complications. C-peptide is not perfect, but it is a direct and reliable measure of insulin production. We've seen today how reliable it is. And as Carla Greenbaum told us yesterday, it measures the disease. It correlates with short and long-term outcomes. We didn't talk about how T1D is an orphan disease. And we don't have a single disease-modifying therapy for this orphan disease. And the patients told us that existing therapies remain burdensome and dangerous. And despite the technology, they can make simple mistakes that can be life-threatening. So we really owe them interventions as quickly as we can bring them to them. So we are now having a very consequential discussion. We are talking about raising the bar above the previous bar. If you recall, the 2009 guidance accepted C-peptide as the primary endpoint for full approval, seemingly. And this is a very important debate. As Dr. Kowalski said yesterday, we are at an inflection point, and whatever is decided by the regulators will result in direct scaling up or down of T1D R&D. So I was very thankful to our colleagues at Novo Nordisk for their candor when they said yesterday, Dr. Gauk said yesterday, that Novo Nordisk may exit T1D if C-peptide is not acceptable as a primary endpoint because of the uncertainty of the conditional and accelerated approval path. We saw today from Dr. Bergold how large and long the studies would need for other acceptable endpoints in the CGM era. They won't fund these studies, phase 3 studies, or phase 2 if they don't see a path to a feasible approval. And we heard about new endpoints, but we all know that will take years to develop. So these concerns are really real, and it is the academic and the industry community's role to help the regulators. The regulators are exemplary in their public service. They care deeply about patients, and we have to help them with these dilemmas. So the message from us, from Provention, is just to try to be flexible and to retain optionality as much as possible. So we want to ask the regulators to please consider the mechanism of the disease. Regardless of the mechanism of the drugs, C-peptide is well suited mechanistically for this disease. It measures the disease. Please consider and help us consider alternative indications. This was something very, I think, constructive that came up of this discussion. Could an option be to use C-peptide for an indication of beta cell preservation? Or Dr. Essen-Moller suggested in the questions for autoimmune insulin deficiency. Dr. de Beaufort said today, the goal is to preserve insulin secretory capacity. So we have a measure for insulin secretory capacity. Or can we discuss alternative methods as we are doing today, borrowing placebos, but also can we talk about key secondary endpoints and not applying necessarily a co-primary? A question for our EMA colleagues. Can we be flexible with clinically meaningful differences, adapted to the different patient populations? Dr. Greenbaum showed us that what matters is the directionality of the change, almost more than any specific threshold. Any C-peptide level retained is associated with clinical benefit. So that's what I wanted to just bring to the discussion is, can we be more practical? Can we focus on retaining optionality? Because at the end of the day, it's going to be risk-benefit, as Dr. Janov and Dr. Moll mentioned. If a therapy is sufficiently safe and effective, we need to find a way to bring it to patients.
So just to give a little bit of perspective on that, and I think appreciate the summarization of the workshop today, Francisco. But one of the things we have heard consistently is that things are based on regulatory frameworks, and the world of approving drugs, although burdensome sometimes, is there for a reason, and regulatory science moves at the pace that sometimes is frustrating, but it is based on what data is put in front of the health authorities. So, appreciating the fact that the consequence of not having endless flexibility or more flexibility can be detrimental to stakeholders who are involved, willing to be able to bring forward assets and et cetera. But it does, I think, come back to that point of saying, well, what is the regulatory framework when we think about C-peptide, and that it can be used as a primary endpoint, but it's not yet a validated surrogate, and the need to be able to demonstrate using the validated measures at this point in time. I think, knowing that is the status quo as it is, wanting to jump beyond it very quickly, but without the data in between is a challenging one. Although I can understand why there's a desire to do so. So, I mean, that's the kind of personal perspective for myself, sitting at the c-path side of things, who sees these in other areas as well. But welcome comment from the folks either on the FDA side or the EMA side to that statement.
Yeah, I'll jump in. We really do understand where you're coming from, but when you use the word practical, can we be more practical? I think that was a euphemism almost for, can we make some assumptions that we just don't have data for in the interest of expediency? You made a lot of statements that were assumptions, I think, that we just don't have support scientifically at this time, and we're looking for the scientific community to help us with that. While C-peptide does represent, let's say, could represent preservation of beta cell function, just hypothetically, there's a couple of other things it could represent. It could, in theory, represent kidney failure and the inability to excrete C-peptide. It could represent a drug whose mechanism of action is to temporarily secrete insulin, such as a GLP-1 analog. But in the long term, we don't know what that could do to type 1 patients. Could it burn out their pancreas faster and at the end of the day lead to more harm than good? And so this assumption that just having a circulating C-peptide that is higher than it was before the drug is not sufficient from our perspective to really confirm that the drug will have the clinical benefit that it purports to have. So these are the challenges that we're dealing with.
Understood, and we completely understand the need to provide additional evidence. I didn't mean that would be the only endpoint to be provided. Apologies if it sounded that way. My comments were more around the level of statistical rigor or the bar that will be applied that would be resulting in very, very large and long studies, as we just heard today, if we require co-primaries and or if we require significance in measures of glycemic control, which are heavily impacted by treat to target strategies and by CGMs, but it's understood.
Yeah, I think that the practical considerations of that endpoint are something that I've come to appreciate a lot in this workshop. And it just as a reminder, FDA does not require that to be the endpoint. We are really open to looking at other endpoints. Something that it's unfortunate we don't have the obvious solution right now, but it's not something that's a regulatory requirement.
Yeah, so I think we are a little bit tougher at this stage. But I do understand the difficulties with the sample sizes. I do know I like the proposal of let's say go into a hypoglycemic definition that is maybe less strict, but if you sufficiently justify that, could be considered as a co-primary, and then the sample size might still be in the order that we would think would be ending up in the feasible setting. Again, if you combine it with a product that is having a relative benign, probably safety profile, that is something there which really could make a case. And clearly that would provide us with some extra information also on the longer term in a controlled setting on other events that are occurring. Maybe we can get observed outcomes, reported outcomes by parents or if possible by the children themselves. So, and that could all together form a dossier for the C-peptide on its own and what the role of it is. I think it would be really helpful for our system, at least in the European context where we are with 27 countries working together. Really helpful if you present this kind of information that we have been discussing the last 2 days in a structured way in a qualification procedure. It gives us a possibility to get different stakeholders on board, patient voice, clinicians from the field, and really have a good discussion, and also looking at these data with our statistical colleagues on, okay, how much really do we think we're here? It might still not end up in a full let's say endorsement of the as a surrogate endpoint, because I think there the issue remains with that we still haven't got that possibility that the interventions are doing different things, and as Lisa just presented, that although strange, these things we have seen happening. So I think that is before we really say something is a surrogate, we really need to establish that, and we may have done that in the past differently, but this is 2021, so the level of science also on our side has progressed, and standards are changing. And finally, I think the unmet medical needs may be there, but then you also must have something that really meets that unmet need. So I think these factors bring in that. And this is also a long-winded response, but I think having certain kind of co-primaries may lead to feasible study designs. That is something individual companies could come with. For C-peptide, I think we really would invite you to come with all the data you have for a more intense discussion with the regulatory system in Europe.
I jump back in for a second. I don't want to monopolize the conversation, but I think it'd be remiss if I didn't directly also address the proposal for the composite of stage 2 and 3 hypoglycemia. And I think that also I'm pretty much open to that at this point. I think it's reasonable to acknowledge that looking at stage 3 alone is just not feasible, and that as long as there's a positive trend on stage 3, that stage 2 could corroborate that outcome. And we are also close to acknowledging stage 2 independently of stage 3 as a clinically meaningful endpoint, in that it's directly sorry, let me correct that, because as Inish said, terminology is extremely important. Not a clinical endpoint, but a clinically meaningful surrogate. So the idea of measuring less than 54 is directly correlated to apparently neurological compromise, as well as perhaps cardiac problems and other issues So unlike less than 70, which is more of a warning value, and we really struggle with that one, less than 54 seems to, if the data that we've seen are correct, to uniformly be associated with adverse outcomes for patients. And so that one, I think that we are on the verge of going ahead with that one. But we'd want to make sure that we have further discussions with our AMA colleagues, as well as really getting a sense of exactly what are the measurement tools we're going to use to obtain those hypoglycemia data, because historically we've used the glucometers for the less than 54, and I believe that a lot of the publications showing that less than 54 is associated with these clinical adverse outcomes are derived from either actual essentially measured glucose or glucometers. And so to the extent we can address some of these measurement issues, just hypothetically, the concept of less than 54 being clinically meaningful, I think we're getting on board with that.
Corinne, did you want to comment?
Yeah, I just wanted to comment on exactly this topic, because here again, I think from my clinical practice, I know that we know very limited about how often 54 to 70 milligram as serious hypoglycemic value is measured. You don't know. And there have been some very relevant studies where they've compared, certainly I'm talking mainly about children, where they have measured and observed the symptoms in IV in a home setting, where it's not that easy to evaluate if you take the finger sticks. So this is something really, I think it's very relevant, like the variability, but it needs to be measured by a continuous measurement device. Otherwise, if you don't measure the right moment, you miss it, and you might miss a lot. And I think that's something which needs to be taken into account if we include this very important measure. And for the hypoglycemia, I think it's valid probably for all, but I was indeed, I would like to quote here the person with the mother with her husband, the adult, where hypoglycemia is the major event, but for the children, there are other very relevant points which need to be taken into account. Sorry.
When you said 54 to 70, I think that I just want to make sure everyone on the panel and the audience we're talking about apples to apples here. From an FDA perspective, again, at 54 to 70 is an alert value. What I was referring to is under 54. So could you just clarify what you were referring to?
I think you need to include all, but even below 54, you can have non-symptomatic and therefore certainly in the pediatric population non-measured, and in the adult population as well. It varies. Symptomatology varies, and if you're only measuring a 24-hour profile, you can have one and you can miss one. So you need to have a longer duration of evaluation.
And also,
and I think
blinded control group, right? Because you would expect probably the the degree of not measuring hopefully would be balanced out by randomization.
Any other comments with this? I don't know if Regina, you wanted to add anything.
Yeah, maybe just I think it's really good to for the field and for the patients, of course, to develop or discuss this, and also, as you also suggest, develop this in conjunction with the regulators, how to provide these data. And of course, as you heard, we do not have the perfect solution either, and this is of course based on what we experienced. But I think that this mentioning of the historic controls, I just wanted to mention one thing that was also mentioned yesterday, this about how to, I mean, that will not solve demonstrating the clinical endpoint efficacy, but there was also mentioned yesterday that there could also be ways of collaborating on registries or something to evaluate these more clinical or longer term endpoints. In that sense, I was just curious as to how you look at that, if there are other opportunities, because of course it's a challenge to select those endpoints up front, as we heard.
Yeah, I think one of the things that we heard from some of the questions that were delivered before the talk, in light of the talks, et cetera, was really, and I think something that has been brought up by several of the speakers, and you're sort of alluding to in some ways, is that as we do trials, it's sort of my way of saying the Heisenberg uncertainty principle. We change the control group when we put them into trials. And certainly when we look at the data that was shown from the type 1 diabetes exchange, we see real world is something very, very different than what we see in these trials, and the benefits would be much more apparent. I don't know if anyone has had thoughts or ideas about how that could be incorporated into this analysis and into these sorts of considerations to try to, again, really get at that real potential of benefit going on. Thoughts from anyone?
An initial thought is it's obviously very interesting, perhaps something to follow on also at a later stage as a understanding of how these drugs behave in a broader population, a more real world population, as you'd like to say. But what you said, and the Heisenberg-Schrödinger comparison, these patients, as soon as they go into a trial, they are different. So that makes the translation to the active treatment group, if you want to use it for borrowing, let's say the placebo, makes it immediately something where all my methodological colleagues get nervous from. I'm personally very, very interested in understanding disease behavior beforehand, having these disease registries that monitor these patients long term, and especially maybe in that setting we can also start collecting the clinical endpoint. But we know also that it's difficult to compare that to the treatment group or to the population that is not treated with the active.
Yeah, for instance, the QR metric is really narrowly defined in that time span and in that study setting where it's right after diagnosis and a randomized clinical controlled trial. So if you're talking about measuring C-peptide in registry studies kind of in the wild, then you get outside the controlled environment from which we presented the QR data from all 9 of these studies. But that really is the framework by which you would screen therapies, right? I mean, it would be in a clinical setting like that, and you would you have that level of certainty in that applied setting. But again, of course, not in the wild, so to speak.
T, I know you were sort of suggesting a relatively smaller size than I think Regina sort of suggested from her analysis. I don't know if thinking about things and incorporating things either way, could we come to something that might address Francisco's concern, which I think all of us have, to the size of trials, and how we could make them a little bit smaller again, whether it's with these other endpoints and the 2 to 3 year extensions that I think we were all talking about over the last 2 days. Your thoughts?
Yeah, I mean the other sample sizes for the larger sample sizes are largely a result of the endpoint being counts or binaries versus the QR metric or C-peptide, which is continuous and it's also adjusted. So by any analysis that's adjusted for things that explain the variability increases the power, and that's what drives down the sample size. But events like hypoglycemic events or binary outcomes typically have less power, require a larger sample size. So in terms of a compromise, the composite endpoints or having multiple endpoints generally also increases power. So you have C-peptide along with the hypoglycemic events or glucose control, et cetera, together as one typically gives you more power as well.
Regina, your thoughts?
Just also, and I fully acknowledge what you're saying, but it's also, of course, again, then choosing the right composite up front, and that can sometimes, of course, also be a large challenge, especially for some of the characteristics of this population, as demonstrated that there's a large variability. So it is, and that's why we wanted to bring those examples, that numbers just get very high based on, and again, of course, that's what we've seen, and different drugs may be different, and may have different characteristics, of course. But but it is really difficult to power up front for those glycemic endpoints. And again, as we also discussed this with the technological development, it might be even more difficult, and we still use the same endpoints for the glycemic control as for the CGM. So that's why I think that this workshop is really good in trying to seek novel ways of doing this. But it is definitely, as we've seen it, the glycemic endpoints that are driving the sample sizes up as high as I wanted to them, and not the c-peptide.
Just maybe a quick comment, if I could chime in on that front. The power calculations that were shown in your talk, T, and others. I think an important consideration is that a lot of these calculations require us to understand the variability in the underlying data that was used to generate those values. And depending on what studies or what data was used for that can have a large influence on these calculations. Looking at a larger universe of integrated data is often really important to make sure that you're accounting for a wide distribution of different types of study design, patient populations, and other pieces of variability that are explainable and unexplainable. But then once you have that large universe of data, which is not easy to put together, explaining as many sources of variability as you can is also critical. So I like the QR metric because it does adjust for a source of variability, and maybe it can be expanded further to account for other baseline prognostic indicators that would help it reduce the amount of variability or explain more variability and get it to be more precise. And then taking that to something like understanding how you're going to power a phase 3, what will those numbers look like? How will they adjust going forward? And we know the FDA, put out a draft guidance actually more recently that it's favorable to adjust for sources of variability to explain differences in treatment effects and to refine that so that as to give the sponsors the most power they have to detect those differences. So I think there's definitely some areas of innovation based on aggregating data that can really bring a lot to the table, answer these tough questions.
So one question that came in across the chat there is along the lines of, based on what we've heard over the last 2 days, there's an urgent need for continued drug development in New-Onset T1D, and the idea of being able to have disease-modifying therapies for patients. And it's important to be able to clarify the definitions of some of the conversations that we're having ongoing today, and to be able to encourage industry to know what the landscape's like when they're making their plans very, very many years out in advance. But with regard to where the regulators are in their current thinking, and historically, the type 1 diabetes guidance being pulled, but not because of anything in type 1 diabetes, but rather type 2, is there a way to be able to communicate the regulatory expectations as these evolve in the future, knowing that science always moves forward, but being able to put pen to paper over time periods? So really giving the opportunity for the regulators to be able to frame that one.
That actually, if you don't mind if I start, just one little comment about the withdrawn guidance. So while the withdrawn guidance did acknowledge C-peptide as a primary endpoint at 1 year, it actually didn't state it was for traditional approval, as it was stated on this panel. That's actually not correct. We never specified the type of pathway that would be used, and the guidance also, while it didn't get into the details about the specific clinical endpoints, it did say that the change in C-peptide should be associated with clinically meaningful measures. So that really is the same. And as Inish pointed out, from a legal standpoint, we were forced to withdraw that guidance because it has some information conflicting with the type 2 safety guidance that we wanted to issue last year. But those recommendations are really not changed from the time of that publication in 2008. I think that we just haven't really had any viable therapies in the space until recently. And so these conversations are now being forced to really think about the practical application of those general recommendations and that guidance.
I think to chime in, I agree. Our guidance and our knowledge on how to deal with these kind of endpoints, for instance, is always based on what we have seen in development and what's happening. I mean, teplizumab is obviously a nice story where we have very promising results already, and where people start thinking, okay, what should come next before we can start evaluating these types of drugs. And I think with that, our guidance is going to be shaped by the developments coming through, by initiatives as you have taken here to say, well, C-peptide is something that is there, with the patient story being elicited, better understanding what they want to see, what this means. But at the same time, we have this basic kind of simple things. Every drug does something on a biomarker that we're looking for specifically, but it goes into a body where there are a couple of receptors around that might be touched by that biomarker as well. Obviously, C-peptide is something that is physical, so we should know a little bit more on that, but the drug does different things as well. And so for that reason, I think it's always with the understanding of different programs, we can come to, okay, well, this is something we trust, and that makes us a little reflective. Obviously, we listen at the same time to new statistical methods that could be applied, borrowing real-world data to perhaps make our concerns smaller by providing possibilities of following patients, let's say post-approval or drugs post-approval in a relevant patient population, or getting some further data in patients that are currently having the disease and that. But I think that's where we are. We are still for this decision making for something that slows down the development of type 1 diabetes once seen, is something where we have no real experience yet.
So what I'm hearing there is in the realization of being able to write down the perfect set of rules with everyone, which they could design their trials around, it's really dependent on what you guys see in front of you on the health authority side, and based on the data that gets submitted to you through the different pathways that are available. So in that knowledge, it sounds like there's an opportunity to collaborate through the appropriate conduits and being able to supply data. And Peter, I know you mentioned on a couple of occasions the qualification mechanism with the Scientific Advice Working Party to do that. So just to acknowledge that, again, I think it underlines the willingness that is manifested by the participation in the regulators here today as well. I'm conscious of time, and I know we're getting close to the close. So, I think where I would summarize on this point, and then we're going to have some closing remarks from Lisa at FDA. But just to say, what we've heard today, again, from the pre-competitive space here across the 2 days, is that action needs to be taken, and everyone wants to push in the right direction. And we're getting closer on the understanding each other's definitions of terms, so that we can plan to be able to move forward together. But I do think it's important to recognize that I don't think there's dispute around the importance of C-peptide as a biomarker in this space, especially in registration studies. But it comes to the complexity of being able to have that validated clinical meaningful measure as well. And I think even if the complexities of HbA1c are true, it's interesting to hear both the EMA and FDA talking about the potential for being able to look at a combined metric to the Novo Nordisk talk for hypo being put forward. That's something that I think being able to flesh out further is something meaningful and potentially very important to the field. But from the other sessions we heard as well, I think it doesn't detract from the idea of when patients' burden is apparent, and we're thinking about being ways to have metrics to accurately capture that. Is it insulin dose? Is it frequency of insulin? Or is it a patient-reported outcome measure or something along the lines of health-related quality of life? I think there's a call from the patients that we heard, the caregivers as well, to accurately describe and objectively measure the burden of the disease on these patients, because that is clinically meaningful. And yet we don't have the tools to describe that in the regulatory construct. So I think there's a gap there when we speak about unmet needs, and what that solution might be to that gap needs to be defined. But that burden of disease is not changing, and it is ever present. So how do we, in the future, be able to do something about that is something I think where I'd seek to be able to discuss with the participants from this workshop and also the panelists through the course of what will write up the details from this meeting report. But on that idea of closing it out, because we're getting time to is close to the clock, I do want to thank everyone for their participation from across all the time zones and putting in all the work up front to be able to record these sessions because of the technical complexities that can arise and I'm experiencing now with my time lag. But again, thank you as well to the panelists, but also to the patients and the caregiver who were able to give us some very focused advice about why we all need to work hard in this. And I think from there, I'll close it out. And Lisa, I know you're going to be giving some closing remarks as well. So thank you all for your time and attention.
Extremely brief. So we probably will be done by 2:30. For this amazing, outstanding workshop. I really learned a lot. Thank you to all the speakers, panelists, and attendees. I was very impressed with the quality of the presentations, the richness of the panel discussions, and the thoughtfulness of the questions posed in the chat. And a particular highlight of the sessions for me was the patient roundtable, which I think was a unique feature of a workshop such as this one. I don't think I've seen that before, and I thought it was really meaningful and moving. A common theme that seemed to run throughout the workshop is that there's much more that patients with type 1 diabetes need, and that we need to work together to achieve solutions. Although we all bring our own perspectives, regulatory, academic, patient, caregiver, what is apparent is that we're all dedicated to making progress. And I do think we are a lot more aligned than not on the state of the science and the unanswered questions that addressing would help move the field forward. And I hope the insights from this workshop will serve as an inspiration and a call to action for further collaboration. You've heard from at least from an FDA perspective, that c-peptide is a useful primary endpoint, but not a validated surrogate to predict a clinical benefit. But we do believe that c-peptide could serve as a reasonably likely surrogate endpoint to support a potential accelerated drug approval, which means that the drug could be marketed while the actual clinical benefit or benefits are being verified and described in post-marketing trials. This approach strikes a balance between scientific rigor and patient access, and without compromising scientific rigor, it strikes a balance between getting patient access with some degree of uncertainty rather than having to know everything at the time of approval. And by making drug development for new-onset type 1 diabetes more predictable and feasible, FDA hopes to facilitate the development of new therapies and promote investment in this therapeutic area. And so to this end, we're open to review proposals for novel clinical endpoints, innovative designs, or other solutions. So thank you again to c-path for putting on a really substantive and polished, from a technical standpoint, workshop. And I look forward to our next interaction. Thank you.